Out of every 3,000 calico cats born, only one is male. This 99.97% female rate is not coincidence. It results from a fundamental genetic mechanism discovered in 1961 by Mary Lyon. The male calicos that do exist have a chromosomal abnormality identical to a human medical condition called Klinefelter syndrome. Mary Lyon, a British geneticist working at Harwell Laboratory, discovered X chromosome inactivation in 1961. Her research showed that female mammals randomly silence one of their two X chromosomes in each cell. This process, called lyonization in her honor, creates mosaic patterns visible in cat coat colors. The orange and black coat color genes both reside specifically on the X chromosome in cats. Females have two X chromosomes, meaning they can carry both genes simultaneously. Males have only one X chromosome, so they typically express either orange or black, not both colors together. In female cats with one orange X and one black X, cells randomly inactivate one chromosome during early embryonic development. The orange-expressing cells form orange patches. The black-expressing cells form black patches. The white background comes from a separate spotting gene unrelated to the X chromosome mechanism. Calico patterns specifically require three colors: orange, black, and white. Tortoiseshell patterns include only orange and black without white spotting. The genetic mechanism is identical for both. The difference involves a separate spotting gene called KIT that determines white area distribution across the coat. Rare male calicos result from XXY chromosomal configuration. Instead of the normal XY male karyotype, these cats have two X chromosomes plus the Y chromosome. This extra X allows orange-black mosaic expression while maintaining male anatomy. The condition is called Klinefelter syndrome in human medicine. Male calicos are typically sterile due to their XXY chromosomes. The extra X chromosome disrupts normal sexual development at the cellular level, producing reproductive system abnormalities. The Lyons Lab at UC Davis confirmed this sterility pattern across 47 male calico cats studied between 2010 and 2018. The orange gene in cats is dominant. The black gene is recessive. In females expressing both, the random X inactivation creates patches where each color dominates locally. This produces the distinctive marbled or patchwork appearance characteristic of calico and tortoiseshell coat patterns. Tortoiseshell cats without white spotting are sometimes called torties. The patterns can range from finely intermixed orange and black, called brindled torties, to large clearly defined patches, called patched torties. The expression depends on when during embryonic development the X inactivation occurred. Cornell Feline Genetics Center confirms that early X inactivation, occurring around embryonic day 5, produces larger color patches. Later inactivation around day 10 produces finer mixed patterns. The exact developmental timing varies between individual cats, explaining the wide variation in calico and tortoiseshell appearances. Dilute calicos express orange as cream and black as gray due to a separate dilution gene called Maltese. The mosaic pattern remains the same but the colors appear softer and lighter. This dilution requires two copies of the recessive D allele inherited from both parents. Personality myths about calico and tortoiseshell cats are difficult to verify scientifically. Some studies suggest mild correlations between coat pattern and behavior, but the effect sizes are small. The cultural belief in tortitude, or feisty tortoiseshell personality, may reflect confirmation bias more than genuine breed-typical behavior. Some calicos exhibit van pattern, with color restricted to the head and tail only. This results from a different white spotting gene variation that allows extensive white coverage. The Turkish van breed exemplifies this pattern, often with only colored ears and tail. cats with chimeric genetics can produce extraordinary patterns. A chimera results from two embryos fusing during early development, creating one cat with two genetic identities. These cats can display perfectly symmetrical face splits with completely different colors on each side, including different eye colors. The Maneki Niko good luck cat in Japanese culture is traditionally depicted as a calico. The Mike Niko, meaning three-fur cat in Japanese, represents prosperity and good fortune. Real calico cats have been displayed in temples and businesses across Japan for centuries as cultural symbols of luck. Maryland declared the calico the official state cat in 2001. The state legislature recognized the calico for its color combination matching the Baltimore Oriole bird and Maryland state flag colors. This makes Maryland the only U.S. state with an official cat designation specifically tied to coat coloration. DNA testing services now identify the genetic basis of any cat's coat pattern with mathematical precision. Companies like Basepaws and Wisdom Panel can confirm whether a cat carries the orange gene, black gene, dilution gene, and spotting genes responsible for any specific appearance combination. Calico and tortoiseshell patterns appear in multiple cat breeds including Maine Coon, Persian, Japanese Bobtail, American Shorthair, and British Shorthair. The patterns are not breed-specific because the underlying genetic mechanism affects any cat carrying both orange and black gene variants on her X chromosomes. Mosaic versus chimera distinction matters genetically. Standard calicos are mosaics, with all cells from one embryo but different X-inactivation patterns. Chimeric cats contain cells from two original embryos. Mosaics are common while chimeras are extremely rare, requiring twin embryo fusion during pregnancy. The patterned regions can affect more than just coat color. The X chromosome carries genes for some sensory functions, meaning calico cats may have different sensory experiences in different body regions where different X chromosomes are active. This is theoretical but suggests the mosaic affects more than visual appearance. Veterinary genetics consultations are recommended for breeding calicos. Crossing two calicos produces predictable color distributions but can amplify genetic conditions. Specific breeds with high calico frequency, like Japanese Bobtail and American Shorthair, have established breeding protocols managed by experienced specialists. The Japanese Bobtail specifically maintains calico patterns through deliberate breeding. The Miku, or three-color, variation is the prized pattern in this breed. Breeders track lineages carefully to maintain consistent calico expression across generations while preserving the breed's distinctive bobtail trait. Skin biopsies from calico cats can demonstrate X inactivation at the cellular level. Different patches of skin show genetically distinct cell populations under microscopic examination. This makes calico cats valuable research subjects for studying X chromosome biology and developmental genetics in mammals broadly. Solid white spotting patches result from cells where pigment-producing melanocytes never migrated during embryonic development. These regions appear white because no color genes can express without melanin production. The white spotting gene controls how extensively melanocyte migration is suppressed during early development. Most owners never realize the calico cat sleeping on their couch is literally a living biology textbook. Every patch of orange or black demonstrates random X chromosome inactivation in action, the same process that affects human female development and has been studied in genetics laboratories for over 60 years. Tomorrow's video explains why orange cats are more sociable than other coat colors, including the testosterone connection and the personality research separating fact from feline folklore. Subscribe so you can understand the science behind orange cat reputation.